So today, let's open the door to a hidden world. We're talking about an invisible ark that's safeguarding some of the most important life forms on our planet, microbes. It's a story of some pretty amazing science, and it explains why keeping these tiny organisms safe is absolutely crucial for everything from our food to our medicine. But what happens when things go wrong? Well, the consequences can be explosive.
Let's start with the question that probably keeps a few microbiologists up at night. Imagine you've poured months of your time, a ton of money, and all this effort into your work. Maybe you're developing a new kind of yogurt or a life-saving antibiotic, and then, boom, disaster strikes. Some unwanted microbe sneaks in, or your carefully grown strain just gives up. The cost isn't just a failed experiment. We're talking a ruined product batch that could be worth thousands or even millions of dollars.
And believe me, this isn't just a what-if scenario. Back in 2016, a brewing company had a serious problem. Their canned milk stout was literally exploding. Turns out the culprit was a wild yeast strain that had contaminated their process. See, it just kept on fermenting the beer right there inside the can, building up pressure until pop. It's a perfect, if messy, example of the high stakes involved in keeping microbial cultures pure and stable.
So yeah, that whole exploding beer situation, it really highlights this fundamental problem, a dilemma that's right at the heart of microbiology.
And here's what it boils down to. If you put microbes in a petri dish with a bunch of food, they're going to do what living things do. They'll eat, they'll multiply, and they'll create waste. But pretty soon they either run out of food and starve, or they pollute their own little world with so much waste that they actually poison themselves. It's a total paradox. To keep them alive, you can't just let them live.
And the solution to that paradox is something called microbial culture preservation. The goal sounds simple, but trust me, it's incredibly tricky. You have to keep a specific strain of microbe alive, totally pure, and genetically the exact same over long periods of time, right? You want the exact same yeast for your beer today that you're going to need 5 years from now.
You know what's cool, though? Every single complex technique we're about to look at, it all boils down to just two really simple strategies. Option 1. Just slow the microbes down. You put their metabolism on pause so they don't eat or make waste too quickly. Or option 2, you give them a new house. You just periodically move them to a fresh environment with new food and less junk.
So let's start with those first kinds of strategies, the short-term solutions. Think of these as the everyday tools in a microbiologist's kit, the things they use to keep their working cultures happy and healthy for days, weeks, or maybe a few months.
Okay, so one really clever method is the stab culture. Instead of just spreading bacteria on the surface of a dish, you use a needle to literally stab them deep down into a tube of solid agar. See, down there's less oxygen and it's a really stable environment, so their metabolism slows way down, and that lets them survive for much longer.
Another cool technique is immersion in oil. You grow a culture on a slant, and then you pour a layer of sterile mineral oil right over the top. This oil basically acts like a blanket. It stops the culture from drying out, and, most importantly, it limits how much oxygen can get to it. Less oxygen means a slower metabolism, which means the culture is preserved.
But what if you need to store a culture not just for a few months, but for years, or even decades? Well, for that, we have to enter the high-tech world of long-term preservation. This is the real frozen ark for the microbial world.
And this is where things get really cool. Literally, it's called cryopreservation. Here, microbes are mixed with a special stabilizing agent, something called a cryoprotectant, like glycerol. And then they're flash frozen in liquid nitrogen at an unbelievable minus 196 degrees Celsius. That protectant, it basically acts like antifreeze for the cells, so sharp ice crystals don't form and shred them to bits. It's pretty much suspended animation for bacteria.
Now, what if you could store something for years without needing an extreme freezer? Well, you can. It's a technique called lyophilization, but you and I probably know it better as freeze-drying. The magic trick here is a process called sublimation. That's where you turn the ice inside the frozen cells directly into a gas, completely skipping the liquid phase.
The whole process is just fascinating. So first, you freeze a liquid suspension of your microbes. Then, you stick it under a really powerful vacuum. This vacuum forces all that frozen water to turn straight into vapor, which then gets sucked away. What you're left with is this dry powder of totally dormant microbes, sealed up in a little glass tube. And to bring them back to life, sometimes years later, all you have to do is break the seal and add a little liquid food.
So, okay, we've seen some seriously cool science, right? From stabbing cultures into jelly to flash freezing them in liquid nitrogen. But why? Why go through all this trouble? Let's connect all these lab techniques back to the real world.
Well, the answer is pretty much everyone, even if we don't realize it.
Medical labs need these standard strains to compare against patient samples so they can give you an accurate diagnosis. Industries, from brewing beer to making cheese, depend on stable cultures to get a consistent product every single time. Research labs need pure, unchanged organisms to make sure their experiments can actually be repeated. And epidemiologists, they store dangerous pathogens so they can track outbreaks and figure out how to fight them.
But the ultimate reason? It might be the most profound one of all. It goes way beyond just industry or medicine. As the source material puts it, the most important reason is the preservation of biodiversity. Just think about what that really means. We are saving the invisible genetic library of our planet, a library that could hold the solutions to problems we haven't even thought of yet.
So I want to leave you with this thought. We've all heard of seed vaults, right? Places that preserve plant species or cryobanks that are saving animal DNA. Well, these culture collections, the ones stored away in freezers and labs all over the world, they are the ark for the invisible majority of life on Earth. And the scientists who maintain them are the silent guardians of a world we are really only just beginning to understand.